Hammy here. Today we're going to start Chapter 8, in which we begin to look at how DNA and the central dogma of genetics and how that relates to what Mendel has said about inheritance and some practical examples that we see in humans and that you'll probably experience throughout your lifetime. One of the first things we want to mention is what we call the Human Genome Project. Okay, the Human Genome Project, once we learned about DNA and we got some techniques and stuff in the 80s, early 90s, in which we could begin to sequence the human genome. So, all 46 of our chromosomes and about all 20,000 some genes. Okay, they began in the early 90s. Okay, 1990, and finished in 2003. So if you do the math, that's about 13 years. Okay, it took them, and they sequenced all 3 billion, that's billion with a B, 3 billion base pairs. Okay, so all the A's, T's, G's, and C's. Okay, so now since that, the last 15 years or so here, they've been working at identifying what that sequence means, trying to interpret it. Okay, what are the genes? What different phenotypes or traits do they cause? And they obviously tend to focus on genes or mutations in genes that cause diseases or genetic disorders. Over here you see kind of a pie chart of what we sort of think are the different parts. And there's some kind of technical language in here, but a couple you might notice. About a fourth of the DNA we think are introns, sort of those in-between sequences that are edited out during mRNA editing. And then just some heterochromatin that's very active in making genes, some jumping genes, remember transposons and retrotransposons, and then some simple sequence repeats that we often use for identifying people. So just kind of an interesting graph here of what we think those 3 billion base pairs, what sort of they contribute to the human genome. When we talk about the human genome, most of those are located, remember we've got 46 of these chromosomes. So quick review is a chromosome is your DNA strands in your cells that are wound around some proteins, okay, called histones. Okay. And that really helps our cell to kind of package, to wind up and package that DNA in a small space. And then as we need to read it, some of it will unwind a little bit to be read and to be made into a protein. Okay. On those 46 chromosomes, the first 22 pairs, okay, so times 2, do the math, the first 44 chromosomes are what we call autosomes. Okay, autosomes are all the chromosomes that contain genes for characteristics unrelated to sex, to male and female. Okay, so if you look at a picture down here of all these chromosomes, you'll see that they've put them in pairs, okay, by size, and that's normally how we do it. You see the first 22 pairs, okay, over to here. So all of these are what we consider our autosomes. And this is a majority of human genes are found on those first 22 pairs. Okay, that last pair we'll look at here in a second. That's called the sex chromosomes, and that determines sex. And there's some other genes on those as well. Okay, but most of our genes are on these first 22, which we say are autosomes, or we say it's an autosomal trait, meaning the genes for that trait are found on one of these first 22 chromosomes. Okay, sex chromosomes is that last pair of chromosomes, and that's what are different in male and female. Okay, females have 2 X chromosomes, okay, like you see down here. Okay, and they're a little bit bigger than the Y chromosome. Males are XY. Okay, that Y chromosome determines male and femaleness. Okay, it has the sex determining region Y gene. So we sometimes call it the SRY gene. Okay, that SRY gene is what, essentially stimulates testosterone and determines sort of all the male characteristics. It's a small Okay, it's a small chromosome. You can see it right here. It's much smaller than the X chromosome. There's fewer than 100 genes on it because obviously they can't be two critical genes because females do not have this Y chromosome. X chromosome can have more genes on it because you can see a male still have an X chromosome and it can have genes that can be read on it. And so this Y determines maleness, and sort of, I guess you could say, female is sort of the default sex in humans. And everyone sort of starts out being female, but as this SRY gene during development kicks on, okay, this will start to change that embryo and to give it characteristics what we determine as male. Okay, and so that happens early on in development as this gene kind of kicks into gear. A lot of times, in order to kind of take a look at a person's chromosomes to make sure there's no disorders, they will take cells that are going through mitosis. So right before mitosis, the chromosomes will coil up real tight and condensed. That makes them bigger, easier to stain, and they'll stain them. They'll take a picture of them, okay, and they'll kind of be all scattered throughout the nucleus. They then either have to cut them out, and we'll do kind of an activity in lab to do this, where you'll get an idea of kind of what a geneticist has to do, and they'll organize them into what we call a karyotype. Okay, a karyotype is that picture of the person's chromosomes. And sort of the illustrations we often use are over here, where it shows kind of the staining or banding patterns of light, dark when you stain them. And they've organized all 23 pairs. Okay, we've organized all 23 pairs. This is an actual picture, like an actual staining underneath the microscope. When you look at the chromosomes, you can see some of those staining patterns match up, and that's why they kind of use these patterns over here. So again, you know, this is sort of the picture type thing, and this is the actual photograph of them. And if you notice, this would be the male chromosome because it has an XY chromosome on it. Okay, and a female would have an XX chromosome. So again, we use these karyotypes to look for chromosomal disorders, missing pieces or extra ones, that sort of thing. Okay, kind of our second topic here for the first part of chapter 8 deals with linkage. Okay, talking about linked genes, and this can mean one of two things. Most of the time it means they're on the same chromosome. Okay, if genes are linked, okay, if this is a chromosome, then there's a gene here and a gene here. If you get this chromosome, okay, if you get this chromosome, you're going to get both of those genes. Okay, so remember Mendel's law of independent assortment said all genes will, or factors or units of inheritance will segregate independently of each other. Okay, that's not always true if they're on the same chromosome. Chromosomes will be passed independent of each other, but if things are on the same chromosome, they'll be passed together. And so the closer two genes are, so for example, down here, this is a linked gene. This is one chromosome. Look, gene 1, gene 2, very close together. They're about always, if you're going to have this trait, you'll probably have this trait too. So a lot of times when we look at people, often people have blonde hair and blue eyes. Okay, so genes that control those are often linked. Okay, if they're on separate chromosomes, okay, then would we say they're not linked? They have an equal chance of sort of going in meiosis during gamete formation of going to separate gametes. Okay, and also they might not be linked very much if the two genes are very far apart on the chromosome. Okay, so the closer they are together, the more likely they are to be linked. If they're really far apart on the chromosome, okay, there's a good chance that during crossing over, remember when they cross over and break and switch during prophase 1 of meiosis, the good thing that there's a good chance that they'll be broken apart from each other. The other thing when we talk about linkage is this idea of being sex-linked. Okay, and this just means that, remember what determines sex is the X and the Y chromosomes. Most of the time sex-linked genes, because the Y doesn't have very many genes, we say that they are X-linked. a lot of times sex-linked traits are on the X chromosome where they're also X-linked. So you kind of might hear them used synonymously if they're kind of, because most of them are on the X chromosome. And it has a unique pattern of inheritance because X-linked X-linked traits tend to be more common in males because males have a Y chromosome and only one X chromosome. So if they get that disorder, females have two X's. So you see here the white is kind of the disorder. So the female will tend to be carriers, but then they have a normal gene on the other X to sort of cover it up. Okay, so they tend to be carriers. And if you look at offspring, you tend to see males have it or be affected with these X-linked traits more often. Things like hemophilia, color blindness, Duchenne's muscular dystrophy tend to be more in males. Females can get it if you have an affected male crossed with a carrier female, then you have a chance of getting two X's with the disorder or with the trait, and then it would show up in a female, but it's much less common. Okay, kind of our last topic here is this idea of mapping genes. And in order to map genes, they actually use linkage. So because remember, the closer they are together on the chromosome, the less likely they are, or the more likely they are to be passed together. So when two things are close together, they have a very low frequency of crossing over, of being split apart in a cross. So that is a lower percentage. And we just use those percentages as map units. Okay, it's not really like millimeters or micrometers or anything like that. We actually use map units, or the frequency of crossing over, to map out or construct these gene maps, or sometimes you might hear them called linkage maps. There was a famous scientist by the name of Thomas Hunt Morgan, okay, that worked with fruit flies, and they only have 4 chromosomes. And so he actually did a lot of work in crossing them and keeping track of the frequencies of crossing over, and mapped out a lot of those 4 chromosomes. And he did a lot of genetics experiments with fruit flies, and they have 6 chromosomes, an X and a Y, a lot like humans do. And so we've used this fruit fly as kind of a model organism for genetics research. But again, similar things can be done in other species in order to construct maps of their chromosomes and where these different genes are for different phenotypic characteristics.